Welcome back to biology. And the last time we were together, we were taking a look at the cell membrane structure. We had seen this image here before, reminding us that the cell membrane is in charge of determining what enters a cell and what can't enter a cell. But we were looking at what the cell membrane was made of. And we learned that the cell membrane was made out of a phospholipid bilayer, two layers of fats pointing in opposite directions with hydrophobic and hydrophilic sections, and then also proteins. And we said proteins come in two different types, channels and receptors. But we never really discussed how they work to be able to allow objects in or out, how the cell is able to make decisions about what is able to enter and what is able to leave. And so this time around, we'll take a look at the function. How does the cell membrane work? One way that it works is to make use of some things that we've already learned about. We've already learned that small molecules can move from high to low concentration through the process of diffusion. And if those happen to be water molecules in this diagram here, well, then we're talking about osmosis. This diagram is showing a lipid bilayer. We do have the phospholipids here pointing in opposite directions, and a high concentration of molecules on one side and a low concentration on the other. And so things that are small enough to fit through the membrane, such as oxygen and carbon dioxide, can freely diffuse from high to low concentration. This particular type of transport is called passive transport. Passive means basically not really doing a heck of a lot. That doesn't mean that there isn't activity going on. It just means that the cell doesn't have to contribute any energy to the process. And so even the dialysis tubing was capable of doing diffusion and osmosis. Dialysis tubing certainly isn't alive. It certainly wouldn't be able to contribute any energy to the process, but it still happens. Passive transport, diffusion and osmosis. But that's great for molecules that happen to fit through the cell membrane. There are certainly objects that are much larger that the cell would want, but if they don't fit through the cell membrane, how is the cell still going to be able to get those items? So what about molecules which are too big to fit through the cell membrane? And this is where those proteins come in. We learned about cell membrane channels, which can bring molecules into a cell opening by forming a tunnel for molecules with the right shape and size to pass. We saw this animation before, where here we have a high concentration outside, a low concentration inside the cell. We do have our phospholipid bilayer, and here's a protein channel. In order for those to fit, they don't just slide in between the phospholipids. Instead, they require this channel protein. And as long as the channel protein has the right shape, then these molecules will then be able to flow through. You can imagine if there is something that was much bigger than these, or if they were shaped like a square or something like that, that they wouldn't be able to pass through this particular channel. That would require a different protein channel. But these are able to slide right through. And if we flip the script and we go where we have the high concentration inside the cell, well, as long as the channel is open, then those same shaped objects are able to pass through from high to low concentration. Now, in this case, because we're not using any energy, then this would be an example, again, of passive transport. Receptors, on the other hand, they work in two different ways. And we learned that receptors, in this diagram here, the cell membrane, and these little curvy shaped items here would be the protein receptors. We learned that receptors are able to grab hold of objects outside a cell and pull them in. That process is called endocytosis. Cyto means cell, endo means inside. And so when we say endocytosis, we're saying that the cell receptors that are on that membrane are able to grab hold of objects that have the right shape and are able to pull them in by wrapping the cell membrane around it. We sort of saw an animation for this the last time as well. We saw this one here where this particular cell was able to grab hold of this green object and pull that on in. And we saw these cell receptors here that had the right shape to be able to bind to the target molecules. And as long as they were bound, then the cell membrane would wrap around it and bring that on inside. Endocytosis. variety of different reasons why this could happen. Sometimes it's about obtaining nutrients. And so here this cell is going to wrap around this large green object there. But other times there's a process called phagocytosis that we'll learn a lot more about next semester. And phagocytosis involves a type of cell called a macrophage. Macro means big, phage means to eat. And there are cells inside your blood called white blood cells. They are large. They eat foreign objects like bacteria and viruses by wrapping themselves around it and pulling those objects on in. In the case of a macrophage, in case of those white blood cells that are eating bacteria, then what they would do is they would send that bacteria to the lysosome, and that lysosome would then break apart the bacteria, beginning the process of helping you get better. Endocytosis can bring molecules in. The reverse of that is called exocytosis, taking molecules out by exocytosis, exiting the cell. And so really it's just the process of endocytosis in reverse. If we go back to the animation and I go backwards, well, that's exocytosis. Endocytosis in, exocytosis out. This type of transport, though, is a special type of transport called active transport. And that's because the cell has to contribute some energy in order for that process to work. These items can bind to the receptors on their own as long as they have the right shape. But once they are bound, the cell still needs to spend some energy to pull those objects in. So this is a little bit different than the cell membrane channels, where as long as they were open, the objects could fit and slide through on their own from high to low concentration. This particular type requires energy. And oftentimes, cells perform active transport to be able to move molecules from low concentration to high concentration. The complete opposite of what we had talked about with other types of diffusion and osmosis. In those cases, it really didn't require any energy. Molecules, remember, move on their own randomly from high to low concentration until equilibrium is achieved. But to go the opposite direction, active transport requires some energy. And so you can think about like a waterfall, that if a waterfall, if you had water at the top of the waterfall, high concentration, then low concentration down at the bottom, the water is going to move on its own. But if you had somebody standing at the bottom with a cup and they wanted to catch that water and somehow transport it to the top of the waterfall, that's going to require a lot of energy. Or in the case of the perfume bottle, when we open up that perfume bottle, molecules move from high concentration inside the perfume bottle to lower concentrations outside. They do that on their own. Active transport would be somebody running around, I guess, with a pair of tweezers, grabbing every single one of those perfume molecules and trying like heck to move those molecules inside the perfume bottle. That would require a lot of work. Active transport overview here. Let's take a look at this other animation. What we're looking at here is what's called a sodium-potassium pump. These are the chemical abbreviations for sodium and potassium. Kind of surprising that potassium is abbreviated with the letter K, right? But there it is. And here we've got the cell membrane. So here we've got a channel. Some channels do work with active transport. And we have a high concentration of sodium outside the cell and a low concentration inside. Well, let's just play the animation first. So in order to move these molecules, these sodium ions, outside against their gradient, then what we have to do is we have to spend some energy. This molecule here called ATP is the energy that the cell uses, and we'll be learning a lot more about ATP next semester. And once that ATP is broken off, then that little burst of energy, that little flash that you see there, go back, play the flash, there it is the energy that causes this channel to change shape. And now we can move molecules from low concentration inside the cell to a higher concentration outside the cell. And so now that it's opened up, the sodium ions can change. It's called a sodium-potassium pump because it can also move potassium ions. Notice we have a high concentration of potassium ions inside the cell, and a low concentration outside. Well, this pump has the right shape to be able to bind to potassium ions too. And so if it's open in this direction, then the potassium ions combine. But in order to move them from the low concentration outside the cell to the high concentration inside the cell, we're still going to have to spend some energy. Again, in the form of that energy molecule for cells called ATP. And so we're going to use a little more ATP this time around. There it is, energy, releasing the potassium ions, again moving from low outside the cell to high inside the cell. So a completely reversible process. But again, active transport requiring energy to move molecules from low to high concentrations. And that's basically it. Cell membranes made out of 2 layers of phospholipids and proteins, but using diffusion, osmosis, and then also channels and receptors, the cells are able to move molecules into and out of cells and control what goes in and out as well. If you have questions, let me know. Otherwise, have a great day.